Topic Notes 11.1 Classification and Embryology. Now it's time for us to start looking at marine life a little more specifically. But before we get started, we do need to understand the basics of classification. And you've probably learned a little bit about this in biology in previous courses. So we're going to go through this relatively quickly, and I'm going to add on a little bit of embryology that's going to help us understand how organisms are classified based on a lot of different pieces of evidence that we have from biology. This picture, by the way, is of colonial tunicates, and these tunicates live in the marine environment, and they're actually our closest invertebrate relatives, which is kind of interesting. And how we know that is based on a lot of the genetic works and the embryological development that we study. So stay tuned, and we're going to learn all about it. But of course, our first big takeaways of this particular lecture, the Linnaeus taxonomic system allows scientists to use a standardized system to identify and group living organisms and to study evolutionary connections. Now, of course, along with that comes lots of learning goals, everything from discussing the function of classification of living organisms all the way down to some of the embryological development stuff that we're going to learn about later on. So once again, remember, these learning goals are things that you are responsible for knowing for future assessments. So taxonomy is the science of naming, describing, and classifying living organisms. So you might ask, why do we need to classify everything? Well, there's a lot of different reasons, but primarily it allows scientists to really be able to talk to each other about different living organisms. And it also provides a way for us to understand evolutionary connections. And this has really come online in modern scientific times with the advent of genetic and DNA analysis. You can just look at some of these organisms on the screen. You have a penguin, which is a bird that swims. Most people recognize birds as things that fly. A dolphin, which we know today is a mammal, just like us. But even today, people still often refer to them as fish. If you look back initially, people would initially say, well, that's a fish. It's swimming in the water. It has fins. So taxonomy had to kind of overcome these sorts of issues. The middle picture is of kelp. Kelp is an algae, yet it looks like a plant. So these are things that we continue to trip over for a long time. Early on, when scientists would try and describe living things, it wasn't always that accurate. Remember, scientists weren't always as well traveled as they are today, and were often working on people's written descriptions or oral descriptions of a particular organism or a dead organism. That doesn't always look like the living one. These are all pictures of organisms drawn back in the 1500s. The left is an octopus, which is actually relatively accurate from 1558. The only really issue is the eyes, which are a little bit more horizontal, but the person doing this drawing was most likely looking at a dead octopus where the eyes kind of inflated out again. And of course, if you look at a rhinoceros, this is from 1515, you're noticing it's missing a horn on the head. It's got a bit of a mustache and sort of a hairy tail. These are things that are not exactly accurate to the rhinoceros we know. But the person making these descriptions in a scientific standpoint may have never actually seen a real rhinoceros. They literally were following a bit of a telephone game from people that had. And Arctic animals like a walrus are even more far removed. The expeditions that actually were going out into the Arctic, not that many were coming back, or if they didn't really have a way to record what they were seeing very well. So as taxonomists became a little bit more sophisticated, we started using what we call binomial nomenclature, which is a two-part name. And this is the genus species name. The first part of the name, or the genus, is the group closely related to that species. We generally capitalize that. The second part of the name, it refers to the actual specific species itself, and this is generally written in lowercase. Now, it is standard also with both parts of the name to italicize them when you are typing. And as an example here, there is a Florida spiny lobster, and the genus species name of that is Panularis argus. So Panularis is the genus, Argus is the species. I like to refer to this like our own names. We have a first and a last name. The only difference here is our last name is usually our group name, our family name, and that is actually put first in our scientific names, Panularis being that group name, and then Argus being the specific name, like our more specific first name. Of course, this leads us to our much larger Linnaeus classification system, which I'm sure you've gone over in biology classes before. Now, Carolus Linnaeus was a Swedish botanist, physician, and zoologist who formalized our current modern system of naming organisms. And he thus was known as the father of modern taxonomy. So in this system, we have 7 hierarchical taxa or levels that we can group organisms in. The largest kind of big umbrella level is your kingdom, and it's the most inclusive. The example would, of course, be the animal kingdom, and of course, the plant kingdom, and such and so on. Below that, we have the phylum level, then the class, order, family, and of course, what we've already talked about, the genus and species. Note those last 2 levels, genus and species, are part of that binomial nomenclature, that naming system of animals and plants and all living organisms. I've also given you an example of our own classification through all of these levels. So kingdom, we are in kingdom Animalia. In phylum, we are in phylum Chordata. We are also in class Mammalia. We are in order Primata. Our family is Hominida, and our genus and species name is Homo sapiens. So we're in the genus Homo, and of course we are the species Sapiens. Now, of course, at first, Linnaeus and other taxonomists of the time grouped species of larger taxa into smaller groups based on their physical similarities. Now, this was a lot of times the best they could do at the time, and they did get a lot right, surprisingly, but of course they got a lot wrong when we are looking at how species are related, and taxonomy sort of evolved into that. You can look at these pictures as examples. Corals on the right there were very much thought of as plants at first because people really didn't know how to really group them. Sponges were another thing. How could this be an animal? It didn't make real sense until somebody really studied them up close. On the left there is our Caulerpa certuroides, or the feather algae, which is a marine algae, but it looks more like a grass or a plant than an algae. So it really took a while before some of these different pieces started to fall into place. Now today we use what we call phylogeny, or the evolutionary relationships among organisms, in order to help classify them. It's called evolutionary classification. And so we organize groups of organisms by their evolutionary relationships and histories. This has caused a little bit of shuffling in terms of classification from time to time as we learn more about the various different living species on our planet. Oftentimes we use things called cladograms, which is a diagram that shows evolutionary relationships. And within that we have clades, which is a group that includes a common ancestor and all the descendants, both living and extinct. If you look at the cladogram on the right, you'll see a bunch of relatively modern animals, everything from a hagfish, which you may not be familiar with, it's a jawless fish, perch representing fish, salamanders, lizards representing reptiles, and so on all the way up to chimps representing pretty much primates. But if you look at the split between the line that would become hagfish and the line that would become the rest of the animal groups, that split represents a common ancestor that they all had. And beyond that split, you see the term jaws. That's where formation of actual bony jaws occurred. So hagfish had shared characteristics of the fact that they really didn't have jaws, and everything before them didn't really have jaws. After that, there were derived characteristics of a jaw. That means all of the other groups beyond that split had evolved and developed jaws. Along the way, you can see labeled out where lungs came into play, where claws and nails came into play, feathers, furs, things like that. So that's the difference between shared characteristics and derived characteristics as you move forward in the evolutionary connections. Now, of course, this diagram is extremely simplistic. We're obviously showing some big sweeping changes just for the explicit reason to demonstrate what we're talking about here. However, I could spend an entire course or multiple courses just talking about each of these different changes and how they came to be. And of course, new data from genetics, from fossils, from all over the place is continuing to reshape the relationships we understand currently about the different organisms and groups within our classification system. So science is continuing to evolve and learn, and we will probably not know all the details ever of these evolutionary connections. We're just doing the best we can with the data we have, and we've come quite a long way over the last 100 years or so. But of course, we want to simplify a little bit simply because we want to be spending more of our time learning about the specific marine groups that you've come to this course to experience. So, but just realize that DNA really has been a game changer in terms of being able to sequence DNA and actually be able to connect these different species together or separate them based on that as well, and really look for that common ancestor. So through this process, we've come up with our modern system of classification and groupings of the various different kingdoms. Now, you probably have heard maybe of a 5-kingdom system. We are now mostly looking at a 6-kingdom system, and this was not only genetic work, but also looking at the various microscopic organisms, and they've actually been a bit of a sticky point in terms of where to put them, especially like Protista and the bacteria and things like that. So we've basically split up at this point based on 6 different kingdoms. We've got eubacteria, or just bacteria really, archibacteria, protista, fungi, plantae, and animalae. Now I'm going to talk about these different kingdoms in terms of this larger 3-domain system that we are now generally operating under. And this separates things out a little bit more simply. You have your bacteria, your archaea, and your eukarya. Now your bacteria, also eubacteria basically, are generally unicellular, prokaryotic organisms and are extremely diverse. Now archaea includes that archibacteria kingdom only generally, and they are, again, unicellular. They are prokaryotic, but they live in extreme environments, and they have specific adaptations that deal with that, which separate them out from the regular bacteria that we generally talk about. Then we have our eukarya, and these are all the organisms that have an actual nucleus, those membrane-bound organelles and all that stuff. The eukarya include protista, kingdom fungi, plantae, and animaliae. Now, as we go through the different invertebrate groups, we're going to talk about their various different classifications and evolutionary connections. And through that, I want to set down a couple of various different concepts so that we can return back to them as we go through all of these different groups. First of all, we can look at what we call embryological development of specific organisms, and through this process, infer ancestral relations. Basically, what this looks like is as embryos develop, they actually show various different traits within that embryological developmental stages that revert back to various ancestral relations. For example, humans, when you are an embryo, you actually develop gills and a tail. Now, those things eventually go away within the developmental process, but they're there. And so that's what we're talking about with embryological development. But we're actually going to go even farther back and look very specifically at the cellular development at a very early stage. So once there's fertilization, you have a little zygote, a little ball of cells. There is a process called gastrulation that starts to occur. And that's where the embryo starts to form germ layers or tissue layers. Now, there are three different tissue layers that we can talk about. There's ectoderm, which is the covering of the surface, that outer layer. The endoderm, that inner layer that lines the gut cavity. And the mesoderm, that layer between ectoderm and endoderm. And that mesoderm generally will develop into musculature and other organs. Now, the diagram included in this slide shows you that process. There's a cutaway where you see that big ball of cells, and you see that hollow space in the middle, the blastocele. Then you see that invagination that basically forms the opening, the hole, and you then start to see the different tissue layers form around that. Now, there are two groups of organisms we can talk about, diploblastic and triploblastic. Diploblastic organisms only form two of the germ layers. Generally, it's just the endoderm and the ectoderm. There's no mesoderm. Triploblastic organisms have all three layers, endoderm, mesoderm, and ectoderm, the whole thing. Now, the images below are an example of a sea anemone, which happens to be diploblastic. They only have two of the layers. So you see the ball of cells, you see the gastrulation happening with the invagination, you see endoderm beginning to form within that, and then eventually towards the right you see the full anemone. Now, this trait of being diploblastic will group anemones and their phylum, which is phylum Cnidaria, with others that are also diploblastic. Similarly, triploblastic organisms are generally going to be grouped together on a cladogram as well. Now we'll dive in just a little bit deeper. Let's label out a few things. The term blastpore refers to that first opening as the ball of cells invaginates and the tissue layers begin to form. That hole enters into kind of a hollowed out area within the ball of cells that eventually becomes the digestive tract. So that hole is a blastpore. Now, there are two different ways this can go, and two different groups of organisms we can give, protostomes and deuterostomes. Now, in groups of organisms that are considered protostomes, that blastpore that initially forms becomes the mouth. All right. And we also say that the early cleavage is determinate, meaning that the cells sort of are determined to become specific things. Now, other groups, the deuterostome groups specifically, the blastpore, that first opening that forms, will actually become the anus. We'll also say that generally, that early cleavage or cell division is going to be indeterminate for deuterostomes, meaning that the cells as they divide up will have a little bit more flexibility in becoming various different types of cells as the organism develops. So all of this being said, we're not going to focus so much on this, but I just want you to have this concept, though, of what the blastpore will actually become, whether it will become a mouth or whether it will become an anus when it first forms, because this will be a characteristic that we will trace through the different invertebrate phyla and also the vertebrate phyla as we go through. So keep that in mind for future reference. All right, that's enough for me. It's time to turn it back to you. So here's your in-depth question for this note set. Sometimes new information or data, like the genetics we've been talking about, can lead to living organisms being reclassified. How does this ability to change help to characterize science? A little bit of a thinker. All right, guys, until next time, keep thinking.